All right, well, you folks are all worn out, and I noticed that the coffee is just about gone. So, you know, I think we're in trouble, but hopefully we'll keep you stimulated. So this hour will be about computational modeling and a systems biology approach, which, from my point of view, I come from an engineering background, and engineering is just naturally thinking about complex systems and quantitation and computation. So even though I'm officially, whatever that means, noted to be in the systems biology field, to me it's just bioengineering. It's a bioengineering take on biology. And so it's fairly natural from that point of view. So what I want to do is give you mainly conceptual view of computational systems approaches in biology, as Roger said, predominantly at the cellular level. But if I have time, there's ways that we have now, and others have moved that up to multi-scale, not just cells, but different cell populations and how they communicate. And if I have time, I'm going to show you a little bit of that too. And I will try to explain some of the mathematical methods, at least superficially, so you get an idea of where they come from, so it's not just magic. But on the other hand, it can't really be a tutorial in all the different computational methods, because then we would be here for quite a long time. So let me start here. To me, if you ask what's systems biology, it really means integration. Integration of multiple things, entities. And I wrote a little editorial for this fairly new journal earlier this year in which I tried to lay this out. If you think about in biology the different kinds of things that need to be integrated, there's really three different kinds of things that need to be integrated. So I've put them onto a three-dimensional plot or a three-dimensional axis. So what are those three? Well, let me start with the one people think about most in terms of systems biology, and I'll call that horizontal integration. And what that means is, let's say you're at the cellular level. You want to integrate at that level, but many, or at least more than one or two, molecular players or molecular components. So that's what people generally think about in terms of systems biology. That instead of studying one gene or one protein at a time, you may study a whole genomic complement. You may do an entire genome sequencing. You may do an entire genome transcriptional profile. You may do whole cell proteomics or phosphoproteomics. Or maybe you only do 10 or 20 or 100, but it's still more than one. So that's what people typically think about in systems biology, that it's horizontal integration of just multiple molecular components at any scale, usually cell. All right, one that's also very important, and people tend to think of really just as physiology, which is fine with me, is vertical integration. And that says whatever your understanding is, let's say at the cellular level, whether it's one molecule or 100 or 1,000 or a full genome complement, you have to know, well, then how does that propagate up not just to that cell behavior, but to multiple cell types in a tissue and to an organ, and then multiple organs, and maybe even a population. So, by my terminology, that's a vertical integration. You're going up different length and time scales. All right, most people would call that physiology. I view that as another component of systems biology. Okay, you need as many molecules at once, and you need to take them up multiple scales. The third dimension that people don't think about as much, but in these 2 weeks that you've had here, I know has been emphasized, which is really good. I've struggled to put a name on it. I've called it operational integration. You could call it maybe functional integration would be good. And what do I mean by that? And what I mean by that is whatever scale you're at, cell, tissue, organ, and however many molecular components you look at, what actually is the information that you're measuring? Okay? It can be as uninformative as merely sequence, the sequence of a gene or the sequence of a protein. Okay? That's kind of informative, but it's not very informative. You'd actually really need to know, for how that system functions at the cell level or at multiple scales, more about the molecular components than just their sequence. You might want to know something about their thermodynamics, or even just their levels, their concentrations. Things about their thermodynamics, their equilibrium, their kinetics, rates of things happening, transport, they're moving around, the actual structure and the physics and the mechanics of how they interact and how that underlies the kinetics and so forth. So I put all of that onto this axis. So when you think about mechanobiology and things like that, you can do it at any scale you want. You can do it with as many components as you want. But what you're really doing is pushing this axis, integrating more operationally or functionally, getting more information about what's actually happening. So do you see that? So when we talk about systems biology and we talk about computational modeling, you want to think hard about what questions am I addressing. Am I trying to learn more about more components at one level? Am I learning how those components propagate behaviors up or down levels? Am I talking about how different types of information are required to be known to pull together? And maybe all of them. So this red arrow, which is time, means research time. And I'll note for Truth in Advertising, Peter Sorger drew this diagram. He's very brilliant conceptually. And what this means is the whole point of systems biology, or molecular, cellular, tissue bioengineering, is to push on all three of these axes. So the very best work would to say, well, we want to encompass more of the key components and do it across multiple scales and not settle for just sequence or expression or kinetics. We want to know more and more all the way up to physical dynamics. All right, so when I talk about the different types of models and problems, you want to put them under this sort of context. And now, again, as to reiterate what Roger said, for most of what I'm going to show here, we're going to live on this axis, because when you think of systems biology, it's mainly including more and more things. Okay? If I have time, I'll show you some stuff that starts to go up vertical integration. And for the most part, I'm quite confident that the last 2 weeks you've learned a lot about this axis. Would that be fair? So therefore, I don't think I need to really add more too much there explicitly.
I'd also like to see the time axis on that, because that's so critical and so often ignored.
Mm-hmm.
You know, how these things evolve in time.
Yep.
Especially in the context of development.
Yep. All right, so that's the context. Now, for what I'm going to spend most of my time on is at the cellular level, because my view is, if we go back to this, if you can't understand what's going on at the cellular level, it's hard to propagate it up too far. You want to do something at the organ level to affect some organ pathology. Okay, your mechanisms are going to end up with molecules inside and outside cells. So if you can't understand it at the cellular level, you're going to have a very hard time doing something else. I'm a big believer in multi-scale models where we get models for what cells are doing, then we put different types of cells together, endothelial cells and fibroblasts and epithelial cells and macrophages. But to create those kind of multi-scale models, what you have to have in that is models for what the cells are doing. You have to have rules that say, well, the macrophages will do this, and then the endothelial cells will do that, right? And then the fibroblasts will do that. But you can't put them together until you actually know what each of them are doing. So it's also the building blocks for going vertically. So that's why we spend, and I think the systems biology field has spent most of its time on this horizontal integration axis. So conceptually, our view is like this, and this is again why I view it as engineering. The quintessential engineering thinking about any problem is engineers are interested in a system, just its operation, and you want to be able to predict it and control it. So there's some measurement of the operation of a system that you want to be able to measure and control, predict. What you have in order to make that prediction and control is the components. So maybe stuff that you put together yourself, you built it out of physical parts or chemical parts. All right, but as you look at all the different component properties, somehow they should work together to be able to predict and control what the system does. All right, well, so we're just viewing cells that way. And the field of systems biology just likes to view cells that way. It says, what's the operation? Well, it's some phenotype. Differentiation, migration, proliferation, death, that's a system operation, maybe all of them at once. What does it depend on? Molecular properties, levels of particular molecules, their functional states, their locations, how they interact, their activities, and yes, it all arises from the sequence of the genome and ensuing structures, but it's got to be characterized with respect to these things, and again, that's that third axis, that operational axis. All right, so that's what we're after. It's a systems biology approach to cell behavior. It's a cellular bioengineering approach to biology, either way. And so in the end, we want to understand what's going on. We want to be able to control it. And you control it in terms of how molecules affect cell behavior. All right, to focus a little bit more, and again, you can cover a lot of ground, but here's the way that I view even what's happening at the cell level. So I already said that we're interested in the response, the phenotype, measurable differentiation, migration. What controls it? Well, what controls it is stuff in the extracellular environment. Might be molecular ligands, might be mechanical stresses, might be radiation, could be a lot of different things. And they control what I'll call execution or effector. So to execute a phenotype, you transcribe the genome and translate them into proteins. This affects metabolism and synthesis, energy production. It affects cytoskeleton and shape and motors. So phenotype actually gets executed by these things. But the execution of these things is governed by the signaling pathways that come in through the cell surface receptors predominantly. So the really unknown frontier is in how the control of the execution processes occurs by these networks. So the control itself, the execution is important, and the regulation of it is important. All right, so for problems like that, we're going to talk about some computational modeling methods, and what I thought I would do is show you a few different examples that come from a spectrum, and this is crucial. A lot of people, especially biologists who aren't engineers or mathematicians or physicists or computer scientists, are very, very skeptical about computational modeling in biology. Tremendously skeptical. Maybe many of you are, and that's okay. Why is that? It's because the vast majority of work that's been offered in terms of modeling biology has been in the realm of differential equations as sort of the classical physics, engineering, mathematics type of approach. And what's the problem with that? The problem is differential equations are beautiful at describing mechanism and dynamics, but in order to pose a differential equation model, you have to know a lot. You have to already know what the important components are and how they interact. And now all you're talking about is how do you now integrate all those interactions. But for most areas in biology, we don't know nearly enough to sit down and write a differential equation model. And so what happens is when we do, and we send it off to a biology journal, they say, well, you don't know this. You're assuming this. You're assuming that. You're assuming that. You've forgotten about this component. You've forgotten about this interaction. And you know, just last month, this was discovered, and it's not in your model. Okay? So, tremendous amount of skepticism, mainly at the fact that we don't know enough. There's not enough prior knowledge to write these kind of models. So, the degree of prior knowledge is a crucial aspect of deciding how to do computational modeling in systems biology. How much do you need to know already? So what the field is doing as a whole, and in our lab, is to say, well, depending on how much prior knowledge we have already, if we're going to undertake a study of modeling some biological system, we're going to have to use a different kind of mathematics on this spectrum, what's appropriate. All the way from knowing almost nothing, and I'll show you an example of this, where you know almost nothing about the system, yet you're going to do some experimental study and you're going to try to get insights and hypotheses and predictions out of that experimental study. What are you going to do? Okay? How do you do something when you don't know just about anything? And can you do fairly abstracted modeling to at least gain relationships about what the important components are and how they affect your system behavior? All right, maybe you know a little bit, you know something about the key molecular players that are involved, and you try to develop a model that says, well, but how do they interact? Who even is involved with each other? Does component A influence component B, or doesn't it? And then other types of mathematics that say, okay, A influences B, how, what's the logic of it? By the time you build in more and more knowledge about how these molecular components interact, you can finally get up to mechanism. So a very interesting process. If you're really interested, let's say you're a pharmaceutical company, and I do a lot of consulting for them, and they want to get all the way to here to a model that's very predictive about the effect of some drug or something, what I tell them, you've got to embark on a process, usually, that goes all the way here and is a couple-year study to say, first we've got to find out what the components are and what really matters. How are they connected and how do they influence each other? And maybe after a couple years, now you can write a mechanistic model, but now you're going to feel really good about it because you're going to have confidence in it because you've built it up. Whereas if you just start from here, chances are you're going to be so far wrong about what's involved that you've kind of wasted your time. Okay, so what I'm going to do is show you a couple examples. An example over here, that's the most abstracted, where we know just about nothing. And the same example, I hope Roger will like it, it's about stem cells. Something about then influences. And then something about logic. I'm not going to say much here about this. Depending on the time that's in, that we have, we could get to an example of that later. But that's the goal. So, number one. So here's the biological problem. And it is a developmental problem. This is embryonic stem cells. It's a study we did some time ago with George Daly, who's now at Harvard Medical School in their Stem Cell Institute. And this was a while ago. It was about a decade ago. And people know more now. But back then, the early part of this past decade, if you took embryonic stem cells and you wanted to know what was going to make them self-renew and make more of themselves without differentiating, or what was going to cause them to differentiate into something else, people really didn't know what the key signaling pathways were that governed that. So here's the study that experimentally we took embryonic stem cells and stimulated them with different combinations of cytokines, growth factors, and matrix. So we would put them on fibronectin, or we would put them on laminin, or we put them on gelatin. And for every one of those coatings, give them either nothing and just whatever autocrine factors they were making themselves, or we gave them LIF, a very important stem cell factor, or fibroblast growth factor, and said, okay, with all these different stimuli, it's going to activate signaling pathways, and that's going to affect self-renewal and differentiation. And from this study, we'll be able to pull out what the key components are, because nobody knows. So the measurement you make is of those stem cells under all these different conditions, a measurement of their undifferentiated proliferation, their self-renewal. A certain fraction of them would proliferate, self-renew. A certain fraction of them at a certain rate would differentiate and become something else. It was very easy in this case to measure the number of cells that either stayed stem cells or had differentiated by this particular transcription factor, OCT4. By flow cytometry, you could measure whether cells had a lot of it, and therefore were still this kind of cell, or if they'd lost it, and some of the cells became this kind of cell, which meant they differentiated. So did this under all these different conditions and all these points in time. because that was the vertical axis, because we want to know rates of self-renewal and rates of differentiation. All right, but for the components then, what are the signaling pathways that were governing that? It was a who knows. So at this point in time, 10 years ago, mass spectrometry to measure things really wasn't very well developed. Some of the modern platforms in flow cytometry at a molecular level and Luminex assays and others weren't really developed either. So all you could do was Western blots, and that's really ugly. Because if you want to say, well, I got 30 or 40 signaling pathways, I don't know which one involved, so I'm going to do Western blots across all of those, and I'm going to do it under all these conditions and all these points in time. Okay, that would have been 5 or 6 years of work, truly, and not very quantitative. So there was a startup company back at that time that had developed through gel electrophoresis some fairly high throughput, semi-quantitative phosphoproteomics. So again, this was pre-mass spec, pre-Luminex, all these kind of things. So we worked with them as kind of a beta test for a few dozen proteins in signaling pathways, because nobody knew which ones mattered, so we just had to measure a lot of them, across all the different conditions, 4 matrix proteins, 4 growth factors, 3 time points. And for all those measurements, for the different conditions and the different outcomes, we did a simple linear regression, a multiple regression of all the different factors, all the different signals, all the different time points against whether you differentiated or underwent self-renewal. And what's shown here are the weighting coefficients. Because if you take a matrix of all the signaling activities under all the different conditions and regress it against all the phenotypic behaviors under all the different conditions, what you essentially get is a weighting coefficient for how much signal was activated under any given condition and how much of that signal contributed to the phenotypic response. And that's what's shown here. What's in the top, the blue and red and black, is for any signal, and each one of these columns is one of the 30-some signals, how much of their signal contributed to some phenotypic response in a multilinear regression way. And all the fancy colors at the bottom were how much those signals were activated by sorting out the different types of matrix or growth factors. So what you should see across all these pathways is, number one, look at all these colors, ups and downs. Just about all the pathways were activated. You couldn't have really picked out and said, oh, this pathway is what's activated. All the others really aren't. We just ignore them all. No, just about everybody was activated. The top plot, with the weighting coefficients of how the activation of any pathway related to the phenotypic response. Is there just one pathway or 2 pathways that stick out here and everybody else has kind of a 0 weighting coefficient because they don't matter? No, just about everybody mattered. So that was a big surprise, because what everybody had hoped is, okay, there's a magic pathway downstream of LIF. When you're on fibronectin, that pathway tells you to differentiate. No. All these pathways were activated in all these conditions. All of them contributed to some extent. There was no magic pathway. You had to include dozens of things. Okay, nonetheless, it's not like you gave up and went home, because what you could do then is decide which ones were at least most important and quantitatively how much they would contribute. So there's a special kind of linear regression called partial least squares regression that it's an inverted look at the problem. What you generally think of in terms of biology is, if I've got some phenotype, in this case apoptosis, and under some conditions I might get a lot of apoptosis, under other conditions I might not. Under different conditions, I'm going to treat with different combinations of growth factors and cytokines, different colors, different levels of that output, and now I'm going to try to match it up with some key signaling pathway. Well, rarely can you do that, because there's no magic signaling pathways. So what you do is you invert the problem, and you say, for all the signaling pathways I've measured, all 30 of them, I plot the cell behavior in 30-some dimensional space. And for any given condition, LIF and fibronectin, no FGF, I'll get a certain amount of proliferation or self-renewal. And it'll plot into this 30-dimensional space depending on what the signaling pathways were. If I have a lot of fork head, well, okay, then under those conditions, I'm going to move pretty far along this axis. And then what you do is you find the key eigenvectors of that matrix. So that's the combinations of those measurements, those signals, that most strongly correlate with the outcome. And each one of those eigenvectors forms what could be called a principal component or a latent variable, but it's a combination of variables. And that combination distinguishes whether you're going to get high apoptosis or high differentiation or proliferation or low. And there can be multiple combinations that all add up, but one or two of them are most important. So it's a way of figuring out, even though everything matters, you figure out what's the most important things that matter, and quantitatively, what do they contribute. And then if this model works well, what's shown up in the right-hand corner is then if you just know the signals that are generated under any condition, you put them into the eigenvectors, and it predicts how much apoptosis or differentiation or proliferation you should get. And then if you compare that against the measurement, you should get a 45-degree line that says, yes, this is the cell's information processing algorithm. This is how it's adding up multiple pathways to give me an outcome. Okay, if you don't get that, then it means your eigenvectors didn't really capture the information content across all those different conditions. So what this means is that you can go in for any one of the signals that you're interested in and see how important it is and how much it contributes. You can actually make a priori predictions. So let's just say in your head, you were an aficionado of the signal protein kinase C epsilon. You've been working on it all your life, and you knew everything there was about it, and you said, boy, I sure hope this is important for stem cell differentiation, because then I'm going to be rich, right? So how much does it contribute? Well, you can do things like this. From the models, you go into your model and you say, what's the weighting coefficient for PKC epsilon? What will it do against differentiation? What will it do against self-renewal? And the answer is going to also be different under all the different conditions, but those eigenvectors will encompass that. You just say, given this condition, how much is this signal? What if I take it out? What do you get? And you can experimentally test it, and the model will predict the right trends. It'll say, okay, this signal is important for this. It's not important for that. So you can actually get the predictions of how much they contribute. And the other important lesson here is, look, the changes aren't that great. Okay, and this flies in the face of normal cell biology. When somebody wants to write a cell paper or a Nature paper or something like that, you want to show a dramatic effect. When I inhibit this pathway or I knock this out, wow, I go from normalized 1 to 10%. Okay? That's not real physiology. That's not real biology. Nothing behaves like that. So how do you get that figure for your cell paper or your Nature paper? You construct one particular assay condition. Oh, I'm going to make the measurement at 16 hours. And I'm going to do it at this level of laminin. And I'm going to pre-starve the cells for 45 minutes. Wow, now I get a dramatic effect. And that's the plot you put in your paper. Okay? And you've demonstrated, yes, it's important. But it's so non-physiological because you've constructed this artifactual assay condition in order to dramatize your result. Okay? Real physiology is these are very complex, they're under lots of different conditions, and you know what, if you inhibit something or overexpress something, chances are you're going to get a modest effect because everything else matters too. So there's actually some important biological insights in this. In terms of computational modeling, it shows that you can pull out the major players and predict what effects they're going to have. Okay, so you can do that, but now you want to do better. Because there was nothing mechanistic about that, right? It just said we made a lot of measurements. We could, by regression, by algebra, relate certain measurements to outcomes. We get the right predictions, the levels of importance. We can predict what happens when we inhibit and so forth. But why? Why are these effects this way? Yeah?
Can you give us some idea, I mean, when you do the principal component analysis, how many do you need in order to capture, say, 90% of the effect on a particular phenotype?
Yep. So there's no theory for this yet. It's all pretty heuristic and empirical. But typically what we found is for the kinds of phenotypes we've studied, apoptosis, differentiation, proliferation, you typically need at least 3 and up to a half dozen pathways. Okay? So you don't need everything. Okay? Rarely can you capture an outcome. Well, you can never really capture an outcome with one pathway. Rarely can you do it with 2. Usually if you have at least 3 of the most important pathways, you get enough information from the network. And usually once you get a half dozen, you're in really good shape. And one thing this will tell you is, let's say in your initial measurement set, you didn't have some of the right pathways, then this model won't work very well. And so one of the questions you ask yourself, why didn't the model work very well? Well, I didn't have the right pathways in my measurement set. And now you have to go back and add those pathways. All right, now let's see if we can do it a little bit better. Can we not only say what are the relationships of the molecular components to some phenotype, how do they influence each other to get to that phenotype? Okay, and here's probably the fanciest mathematics that I'll show you. It's reasonably complex, but I'll try to make it intuitive. All right, it looks something like this. Now again, in biology, cell biology, biochemistry, you draw a lot of qualitative models like this, don't you? You say, well, I've got EGF receptor, and it goes to ERK, and ERK affects this transcription factor, but there's some crosstalk from AKT, so you draw lots of things like this. All right, and that's good. The problem is when you just draw them, you can't do a calculation on it. Okay, if this is complex enough, you can't say, well, but what if I overexpress this and I have an inhibitor against that? How do all these add up? There's no quantitative calculation you can do. So what you'd like to do is still get these kind of graphical models that you can look at and kind of make some sense. But you actually want them to be computable. You want to be able to put numbers in and say, okay, if this, then that. So what you want to do is to turn a graph like this into what are called conditional probability distributions. So for instance, let's say A and B are both receptors and C is some particular signaling molecule. So if receptor A is active and receptor B is active, that's 1 and 1. Well, it may well be that I've got a probability then of 95% that this won't be activated and a probability of 5% that it will. But if I take away receptor B, now there's a big probability that this will be activated, and I can quantify that. And they're generally probabilities because there's noise in data. The noise might just be either technical measurement, or it could be variability. And if I'm averaging over multiple cells, then maybe 90% of them are doing one thing and 10% are doing another thing. And these probabilities will capture the heterogeneity rather than technical variability. That's a notion you can test. Okay, so that's the idea, to turn these qualitative pathways into quantitative pathways, models. So how do you do it? So you take that same kind of data. So remember, under all our different conditions, measurements of 30-some different pathways, all the different activities in them, and measurements of differentiation and self-renewal. Okay, how do you now generate a whole network that has things upstream, downstream, and quantitative effects? Well, it's really this. And in essence, it's a very crude and straightforward calculation. How you actually do it is where all the complication comes in. It really says something like this. If I have let's say I have 4 components, I could imagine different models by which they affect each other. I could imagine a nice simple model where the activity of 1 only affects the activity number 2, and 3 only affects the activity of 4, but 2 also affects 4. Okay? That's one model. You can imagine, oh, yeah, that's true, but 1 might also have an effect on 3, and 1 might also have an effect on 4, and 2 might also have an effect on 3. So how would you choose between these models? Well, in essence, all you do is you try them all. Okay? So a Bayesian model basically says, if I want to know the probability that my model structure has matched my data, okay, I end up calculating across all the different models which ones match the data this well, this well, this well, and that well. Okay? computationally, there's some very nuanced tricks for how you do it. But don't let anybody fool you. You're really just getting best fit models. Okay? Of all my components, how do I best explain my data? It's kind of like if you're familiar with differential equations and you have parameters and you simulate the model and you say, okay, if I increase this parameter, I fit the data better. It's really basically that. And here the parameters are, do I have an arrow from 1 to 4 or don't I? I fit the model with it, I fit the model without it. If I have an arrow, okay, and it also depends on 3, well, is it 90% of 3 is on and 1 is on? Is it 44% of 3 is on and 1 is on? Okay, and you just have to calculate all those probabilities. Okay, that's not really how you do it, but that's really what's going on. So it's just a best fit out of all the possibilities of who affects each other, okay, and how well that fits the data. Okay, so it's really not magic. And what the computer scientists argue about are the algorithms by which you find that best fit. But that really doesn't matter here. All right, so for the same data set, we can run that sort of a calculation. So these things, so in these purple outlines are the cytokines and growth factors. So here's LIF, here's laminin, here's FGF, here's fibronectin. So those are inputs. So there's no arrow that goes to them because you have added them in this case. These triangles are the phenotypic outcomes. The rate of differentiated proliferation, the rate of undifferentiated proliferation, the rate of conversion from a stem cell to a differentiated cell. So these are the outputs of the responses. So they don't affect anything else in this construction. So there's an information flow from the purple circled things that are the stimuli going down to the different responses that are the phenotypes. And then what you want to know is what are the molecular signaling players in between and how do they affect things. So what that model does, out of all those activities that I had shown you back here, okay, now we try all the possible graphs and what fits them best, and we get something like this. And says, well, okay, activation of LIF affects the activation of STAT3. That affects the activation of RAF. That goes down and affects, this is another phenotypic outcome I won't talk about. This also affects ERK. ERK affects GSK3. That affects a different ERK. That's also input by FGF. Okay, this affects SARC. That affects, oh, I forget even what NR1 is at this point. This affects PKC delta, but PKC delta doesn't actually have any effect on my 3 phenotypes. Why is it there? It's probably affecting some other phenotypes, but it's not affecting the phenotypes I measured. So here it's a dead end because it doesn't affect any other phenotypes. But you see the way that it goes. Okay, you can actually construct, and each one of these arrows now is not just a qualitative arrow, it's actually a number. There's conditional probabilities of the probabilities that if this is on, and that's on, and so forth. Don't know if I have a picture of that here. Okay, yeah. So that's an interesting thing about Bayesian network models. Formally, they're called directed acyclic graphs, and you cannot have a feedback loop at any one time point. Now, of course, there are feedback loops in biology, so there's different ways to do it. One thing you can do is just do different sequential Bayesian networks. So let's say I've got time points at 6 hours, 12 hours, and 24 hours. Okay, I could develop a model at 6 hours. Then I could develop a model for how the 6-hour data affects the 12-hour data. I could develop a model for how the 12-hour data affects the 18-hour data. And so the 6 to 12-hour data might have an arrow going in this direction. The 12 to 18-hour data might have an arrow coming back that direction. That's one way to do it. Of course, that only works if the feedbacks are on separate time scales, right? That something's happening sooner and something else is happening later. So how you build in synchronized feedbacks in Bayesian network models is actually a very interesting mathematical challenge right now. So there's some people proposing to do it. The main problem is that the data you need is really daunting. Because in order to do that, what you have to have is very specific inhibitors. And say, okay, if I'm able to inhibit this, okay, now am I affecting something, what's putatively upstream or not? Okay, and right now, people haven't generated the kind of data sets you need for that. So right now what people generally settle for is the sequential models with feedbacks presumed to happen at different time scales. So that's one of the weaknesses of this kind of modeling. And other kinds of logic modeling and things too. That's one of the challenges. Good question. Okay, so my point here is, remember the partial least squares regression predictions that predicted the effect of PKC epsilon on something like the differentiation, the conversion of stem cells to differentiated cells. Okay, by the partial least squares modeling, it was just a quantitative correlation. We said, how much did it contribute? Well, now you see the pathways, okay? Some things work together to activate PKC epsilon, and it does some things that go downstream and affect these rate constants for differentiation or renewal. Okay, so you get to start to see some of the information flow. Now, what I don't know is every time I draw an arrow, I don't know if this molecule is actually interacting with that molecule directly. Is it docking? Is it phosphorylating? No, there's no mechanism here. But I know topology and I know influence. So now you could push it deeper, which we didn't do, but you go back and say, all right, now let's do some really dedicated biochemistry experiments and say, well, how does PKC epsilon affect PKC alpha? Okay, what's the substrates in between and so forth? And then finally you can get to a differential equation model. Okay, that's the idea. Okay. Okay, I was wondering if I had some of these. So I told you that these arrows now actually represent something quantitative, right? They represent these sort of conditional probabilities. What's the probability if this is active and that's active, that these are active? And you get those out of the model. You don't just get what the key arrows are. You get quantitatively what they look like. And these can be kind of interesting. So for instance, here's one between, on this axis, if I just look, this is the activity of SARC. And this is the activity that PKC epsilon. And where I have green, that means there's a very high probability. So what does this tell you? What this tells you is there's an inverse relationship between SARC and PKC epsilon. That activity of SARC would depress the activity of PKC epsilon because I have a high probability when I have high activity of SARC to have a low activity of PKC epsilon. Okay? It's green here where I have not much activity of SARC and more activity of PKC epsilon. So that's a kind of an inverter signal. Here's an interesting one. When PKC delta is on, okay, that's off. When this is on, when this is off, that's on. And it's not sort of a linear inversion. It's just sort of a yes-no logic gate. So the logic, it's still repressing, but it's a different kind of thing. There's some that go the other way. Here's MEC and GSK3 beta, and now the green goes this direction. So the more activity of this you get, the more activity of that you get. On this side, they're more complicated, where I have two things upstream and affecting the downstream one. So I won't take the time to explain this. But you can quantify the effects of the upstream factors with the downstream factors. And that's very powerful, because that allows you to make quantitative predictions. So if I say, what happens if I have an inhibitor against this? Well, you can run through the calculations and say, well, okay, shift the probability then of my differentiated proliferation by this much. Okay, so that's a neat thing. And it also tells you, and I think this will work well, that a logic model might work pretty well. Okay, and this is where we're spending a lot of our time these days is casting these signaling network models as logic models. And one reason we've decided that's a good thing is because these relationships were totally empirically gained. We didn't say, okay, we're going to assume that there's a positive relationship between MEC and GSK3 beta. We do the experiments, what's called data-driven modeling. The data tell you what the relationship is. But what comes out is there are logics between the activities of upstream things and other things and one pathway to another pathway. So it says, well, we could probably write a model that's not this ambiguous probabilities, but just says, okay, there's going to be an AND gate or an OR gate or something like that, and we might be able to fit the data pretty well. Because the cells themselves, their biochemistry, as empirically measured, is telling you that there's actually logic in these circuits. Not because you said so, because it's a man-made notion, but because the biochemistry is telling you that. Okay, So that's the last piece, is then what if we explicitly do a logic model? So now we're assuming a little bit more. We're actually assuming that we know what these interactions are and that they're going to operate by some kind of a imposable logic. It's just a matter of calculating what that logic is. With the Bayesian networks, we didn't assume that. We just threw everything on the table and allowed these things to emerge. Okay? So that means we'll get more bang for our buck because we're assuming more prior knowledge. And this is good. It's very timely in systems biology because the things I've told you about before really said we have no prior knowledge. Okay? But there is. It's not enough generally to write differential equations for mechanism, but there is some prior knowledge. What's that prior knowledge? It's living in all these databases. Okay? There's dozens and dozens of interactome databases, right? And pathway databases. So something like this. This is Ingenuity. It's one of the most famous. It's not necessarily that it's good or better than anything. But this is a picture that comes from a pathway database. So what is this? I'm going to talk about the last example we'll have to do with liver inflammation. But what this is what the databases say. Signaling networks are downstream of different growth factors and cytokines, TNF, interferon, TGF, IL-1. Turns on these signaling pathways all the way down to transcription factors that affect proliferation and death and cytokine secretion and so forth. So if you go into Ingenuity and you pay the money and you say, tell me the signaling pathways from these cytokines to these transcription factors, you will get this diagram. So it's not as if nothing's known. Okay? From curation of lots and lots of literature, all these kind of interactome pathway databases will give you these kind of pictures. Okay, what can you do with them? So this is prior knowledge. Okay? It's not good enough in itself. I would defy anybody to just sit down and now write a model based on this and try to predict what's going to happen. But what if we took that sort of a pathway database and turned it into a formal logic model that, again, was kind of indicated by the way the Bayesian networks came out. And we said, okay, well then, let's propose that E is active if A is active and B is active, and then F is active if C is active but not B. Okay, we can now very explicitly cast models this way, superimpose them on this database, and what? Well, you have to match them to data, right? So let's match them to data. So let's do a similar type of experiment. In this case, this is hepatocytes under inflammatory conditions. It's not embryonic stem cells, but this has happened to be the study we did for this. So treated primary hepatocytes or different liver cell lines with different cytokines and growth factors, measured activation of transcription factors and cytokine production and apoptosis and proliferation, and measured a lot of the signaling pathways in between. Yes?
So is this only for like phosphorylation networks, or does it hold for transcription factors and expression?
Anything you want. And in fact, if I get to that, I'll show you how it connects all the way down to transcription factors and beyond, because that's very important. Okay, so you generate data like we did for the embryonic stem cells. So this just happens to be, let's just look at the left. This is primary human hepatocytes. Each of these columns is different growth factor or cytokine treatment. We also put in pathway inhibitors, all the little tiny columns. And each one of these rows is one of the signaling activities. And so you can see whether they're transient or late or sustained or nothing at all. So you get this kind of data set. And now here's the idea, is you map it against the database. So you say, I start with some pathway map that's imaginable from prior knowledge literature database. And now I'm going to turn it into formal ands and ors. And I'm going to run through all possible combinations that I think are plausible. Okay? It's not as many combinations as Bayesian networks, because Bayesian networks can be any kinds of interactions with any kind of quantification. Here, they're much more strictly enforced. It's an AND gate, it's an OR gate, it's a NOT, and so forth. But now I'm going to map it against all that experimental data and see what comes out to be the best fit model. So the concept is the same. It's just a simpler mathematics. And I can fit the model both by the deviation from data. I can penalize it for having too many arcs, too many components. You might think, I'll fit the data better if I add more components and add more interactions. Turns out that's not true. Turns out the more components and interactions you get, the worse you fit the data after some optimum, because you're introducing false positives, things that really aren't there. And so, just to show that you start with just an initial pathway map from the database, the yellow and red mean you don't fit well. You now train this logic model, and there's very little red left, and you fit pretty well. You can predict now a priori effects. So if we add new drug inhibitors or new combinations of cytokines and just a priori run it through that logic model that's already been fit, and you can predict pretty well a priori what the different activities are. Okay. So one thing you can do with this is compare differences between, let's say, primary hepatocytes and tumor cells. So where they're black, they're the same. Where there's red and blue, there's differences. Because you might want to ask, well, what if I got a normal liver? What if I got liver cancer? Where should I drug? I want to know what pathways are different. So you can do that. We've done that in great detail. And in excruciating detail, we've said where are there pathway differences between primary hepatocytes and the liver tumor lines, and what's different in the logic. For instance, this one's really interesting here. Downstream, there's the NF-kappa B pathway that's really important for cytokine production and survival. And in normal hepatocytes, you need signaling both through the insulin pathway and through TNF. And you have an AND gate. You have to have both these activities and turn this pathway on. In the liver cancer cells, they're just totally indifferent to this pathway. It's either already activated or you just don't even need it. All you have to do is come through this pathway and on goes your NF-kappa B. So you can very specifically figure out where the biochemical logic is that's different. And I won't go into it because of time, but we've been able to validate this in terms of potential therapies, which kinase pathways are actually different between normal and diseased that might be useful in hepatocellular carcinoma. Okay, let me stop there. Oh, I was going to go, yeah, then all the way to the transcription factors. Let me just quick put that up. That's what I was hoping I might have a little time for. Where is it? Yeah, so instead of just living on phosphoproteomics, let's say in the same type of experiment, we've done that. Here's now the signaling pathways. So for all sorts of different stimuli, the different pathway activities at the phosphoproteomic level. Now also you measure gene expression. And so for thousands and thousands of genes, you get what's up and what's down. Okay, how do you connect the two? Well, with Ernest Frankel, who's really a genius at this, you have to figure out which transcription factors are responsible for which gene expression. But there's ways to do that from motifs and from where the DNA is open with respect to its chromatin, hypersensitive regions. So you identify transcription factors that are responsible for the gene expression. You connect those transcription factors to the signaling proteins through the same kind of logic network. And it looks something like this. So here, all the blue are the measured signaling proteins. All the yellows are transcription factors. These green folks up here are the stimulatory ligands. So you can go all the way through seamlessly. And there's one big logic model for stimuli, signals, transcription factors, and then we've actually measured cytokine release too. That's governed by the transcription factors, and we connect that as well. So that's one nice thing about the logic modeling is you can seamlessly go across what's called heterogeneous data, because you haven't imposed physical mechanism. Okay, so that's very nice. All right, I think I'll stop here and just say, yeah, let me just leave it at this. So everything I've told you for the last 45 minutes has been for like one cell type. All right, and that's good. You need that. But if you're going to do this in tissue, you need it for all these cell types. So if I wanted to have a model like this that has something to do with vascularization and stem cells and how they're affected by monocytes and macrophages, what I need to know is, for any given cell type, when it's got stimuli in its environment, what's it going to do? Is it going to die, proliferate, secrete cytokines? When it does, Now that affects what it sees, it affects what the other cell sees, so now I got to have a model for this one. This is when it sees these cytokines or growth factors, what's it going to do? It's going to migrate, proliferate, differentiate, secrete growth factors and cytokines. Okay, they're going to feed back on him, they're going to feed back on him. So I've got to have all these operating simultaneously. So I need a model just like I showed you, logic from growth factor cytokine matrix in the environment, down through signaling, through transcription, cytokines, apoptosis, differentiation, and then connect that up through the extracellular mediators to each of these. So this is a complicated task. Roger and I, in a collaboration, have an interesting start on this. We've now, in my lab and Kevin Haggis at MGH, we've started to do this in animals. So the very kinds of measurements I showed you that we've made in cell culture, we can actually make them in mice. And with Linda Griffith, who was just here, we're actually doing some of this in clinical patients. What kind of tissue and fluid samples can you get from clinical patients? Do the same kind of measurements on different cell types and cytokines and signals and put them together in these kind of overall models. So I see a very, very promising future. The concepts are there. And more than anything, it's a matter of what experimental conditions and what experimental samples you have access to. I think the computational frameworks are there. Okay, I think that's the 10:30, and now we can have lots of discussion. If you lead to things, there's a few more things I can show you, but we'll see if you want to get there. Good? Yes, sir.
So in the earlier slides, when you're only studying sort of a subset of all the possible signaling molecules, is that how, from what you just said, you pick those, what you look at based on what's available to you and what you're interested in?
Yeah, that's a great question, because that's most important, because you know, what's an aphorism? You can only model what you measure, really, okay? And if you don't measure the right things, your model's not going to be very useful. Okay, so what's the approach to that? Sometimes there's enough prior knowledge to say, well, look, if we're dealing with proliferation of fibroblasts, okay, we kind of know. There's these growth factors and this ERK pathway and JNK pathway and so forth. There's so much in the literature already, so much prior knowledge. We have a good idea what to measure. We measure that. We develop the computational models. And now you ask, okay, can we now make predictions? And for the most part, they work pretty well because the prior knowledge was strong enough to do that. Sometimes you can't. You put in some inhibitor and something weird happens. And you say, aha, there's something we didn't know. We needed to measure this pathway. And because we didn't measure this pathway, we couldn't quite get all the effects. All right, so that's the easiest case. You know enough to get a pretty decent model, and then just by pushing the model predictions, you find some places where you don't have it quite right. Okay, the other extreme was like the stem cell one, which again, back then, nobody knew anything. So you try to cast as wide a net as possible. Now, how wide a net can you cast? Okay. Well, for most kinds of biochemistry, you're restricted by antibodies that you have. Do I have an antibody against this, 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 and that? Now, of course, there's gazillions of antibodies. And so one approach that some people have been using is what's called a reverse phase lysate array. I don't know if anybody's heard of that. You actually take your tissue sample and you just spot it thousands and thousands of times, and then you come in with some antibody against this phosphoprotein or this phosphoprotein. Okay, it's not a Western blot, because with a Western blot, remember, you first make sure you get the right size in your column, and then you come in and get the right size against the recognition antibody. So the problem with reverse phase lysate arrays is they're usually wrong, because there's nothing specifying that you really have the protein that you have. Yes, you have some recognition antibody, but there's so much cross-reactivity that it could actually be telling you there's this other protein and this other protein and this other protein. So that's why the best new high-throughput methods combine the specificity of Western blots with some high-throughput thing. Luminex is something we particularly happen to like. It's essentially flow cytometry, but it's essentially a Western blot on a flow cytometry bead. You actually have a capture antibody and then another separate independent recognition antibody. So there's a much greater chance you're actually measuring the protein you think you've got compared to a protein array. Okay? The problem with that is even though there's thousands of antibodies that you can put on a protein array, there's not that many for which you get both a good recognition antibody with an independent epitope against a capture antibody for the same protein. Okay? Turns out, you can go out and we do this for a living. Try to measure all the signaling pathways you can, and for many of them you can't find actual reliable pairs of capture and recognition antibodies to really know that what you've got. So, of course, what you can do is generate your own. Okay, I got to have one for this pathway. Well, that can actually be quite arduous and can really develop new antibodies and get all the right epitopes, but it needs to be done. So, what's the other possibility? Mass spectrometry. And I mentioned this before. Mass spectrometry, you can measure everything in principle. Okay, and the very, very best labs at this, you can just put a sample in and essentially measure every protein that's there. And by getting some calibrated mass and charge tag, figure out what it is. And you can get hundreds and hundreds and hundreds of proteins. What's the problem with that? It's not high throughput. It takes forever. You know, you do that across maybe 3 different conditions. Well, look what we're doing here. 16 conditions, 30 conditions, 50 conditions, right, to get all the possible stimuli and concentrations and whatever. You cannot do that with mass spec. It's just so low throughput. Okay? So you can back down a little bit and say, I'm only going to measure phosphotyrosines and not everything. Now that's higher throughput, but now you're missing a lot of things. So those are the trade-offs. If you want to capture everything, you really can't. You're making some compromises. If you get very reliable about what you're measuring, then yeah, you're going to miss a lot of things because really you don't have reagents for them. It's a real conundrum. So the breakthroughs for this are going to continue to be technical with respect to reagents and biochemistry measurement technologies. The math is ready. The math can handle all of this. It's can we really get those measurements? Yeah.
I just wanted to relate the 3 methods you talked about, the regression method,
Yeah.
the Bayesian method, and then the logarithm.
Yep.
So in the regression, you talked about, you go ahead and you find the eigenvector that necessarily tells you that all these pathways are orthogonal independent. Then you go to Bayesian and then they're all talking to each other, right?
Yep.
And then you have probabilities in there too. And going to logic, you take the probabilities out.
Yep.
So how are they all related?
They tell you different things. And that's why I really like that slide that has the spectrum on it. I think it's really important. They tell you very different things. So to start up here, when we explicitly impose logic, they will tell you what is the logic because that's what you've asked for. You say, I want to know, is A and B an AND gate or an OR gate on C, or no effect at all? Okay. With this, it's not quite as good as that because you're letting the influences be whatever they are. And even though sometimes it'll look like a pretty clear logic, and I pointed out some of those. Oh, when this is active, that's inactive. For many of the other interactions, it's kind of a mess. I got some, this is probable, and this is probable, and this is probable, and it's really hard to wrap your head around the intuition of that logic. So you've got some quantifiable influence, but it's not really an intuitively interpretable logic for part of this. Okay? For this, you have no idea who's influencing anything other than who's correlated with something else. So you can get correlations of some pathway activities with other pathway activities. You can get some correlations of some pathway activities with a phenotype, with a transcription, with a metabolic activity, but it's all correlation. Okay. And so it tells you which correlations are positive, negative, strong, weak, but that's all you know. So my advice is for any lab that would like to be a computational systems biology lab, you view this as your toolkit. And and other things that I haven't mentioned here. But you got to think about having a large toolkit. It's kind of like being an experimentalist. You would say, all I do is flow cytometry. Okay, that would be a pretty weak experimentalist. Yeah, no, I actually can do some other types of experiments too. So same with modeling. Yeah.
To what extent do you think it's possible? I mean, you talked about these different modeling approaches independently.
Yeah.
Are people trying to develop platforms where one level sort of informs the next, or they communicate with each other and there's interaction?
Yeah. Not that I know of. That's what I'd love to do. And I wrote a grant proposal to a pharma company a year or so ago that was interested in that. Because what they said was, well, for some potential markets, some diseases, we know that in the end, we want this kind of a model because you got to go in and say, okay, I think this drug has this mechanism and this is what's happening. But, yeah, for just about all the diseases they're interested in, they can't go there. And so what I pitched to them is, well, let's just do this. And it'll take us 3 years or 4 years or something like that, and 2 or 3 postdocs, it'll cost you $1 million. But you know what? $1 million later, you'll have moved a whole disease from having no idea where to go up to something like this. And you'll have a platform inside that you could use this for all the diseases. In the end, they decided they didn't have enough confidence in that for the amount of money that it was going to cost. You're not going to understand that. I can't prove to them that it's going to work. It's a research thing. So, what's conceivable is you could imagine an NSF or an NIH grant on something like that to say, we're going to take a really important biology problem, in which just about nothing is known, and we're going to march up what I call the modeling food chain. And it's going to be the same biological system. We're going to start with one set of experiments, do one kind of modeling, then what other data do we need to get to move it up, then what other data do we need to get to move it up, and then we focus in and pull it down. What's the key logic areas? What's the key place to do mechanism? And we're going to go all the way up to some really predictive drug effect. Yeah, maybe we should write a proposal like that. Okay, there's nobody I know of that right now has turned this into a platform. I think it's just a concept. So we've done bits and pieces.
About the Physiome project, mostly in Europe, but I guess that's primarily at sort of the differential equations model level. I don't know to what extent they've done.
Yeah, and I think, I might be wrong, you know more about it, I think they're focusing a little bit more on vertical integration, and then don't capture quite as much this way, although I know they're someplace over.
Right.
So. So I think it would, it's interesting. It could fit into that to say, why don't we build in some of this horizontal integration into this machine that's already focusing on vertical integration? Yeah. There was one, yes.
For the same thing, sometimes we have dependent and independent variables. So where we consider it in a model, or if I want to consider it separately, the both dependent and independent, so where I can consider these?
That's a great question, because in a lot of these things, I don't even know necessarily what to look at. We'll go here. A lot of things they're both, right? That some of these activities, some of the phenotypes are both dependent and independent. So when I showed you the Bayesian network model, I said something like, okay, there's no inputs to how much laminin I have. And at the end, this cell response is where I stop. Well, of course, that's not true. The cells might be making their own laminin. Now, is laminin an independent variable or is it a dependent variable? It's both simultaneously. That goes to the question earlier about feedback loops and time scales. So one nice thing about both the logic modeling, and the Bayesian network modeling and the regression modeling is you have the opportunity to place any measurements anywhere you want in that. You can say, I'm going to treat it as an independent variable that's affecting some other variables. I'm going to switch it around. I'm going to see maybe it's actually predicted better as a dependent variable from these others. Right? So you actually can do that. You can move variables around to be either independent or dependent and see where they actually play a more reliable role in the model. Yeah, because in biology, yeah, everything is both simultaneously independent and dependent. Yeah. Yeah.
Especially with the regression, what level of significance did you use for the variable, and also why?
So what we generally do with regression is a lot of statistical cross-validation. So we'll randomize blocks, we'll leave some data out, predict that. And so what we generally look for is a level of significance that when you do the randomizations that you haven't introduced noise that makes the cross-validation capability worse. Okay, so it's not as if it's a magic number of p less than 0.05 or something. It's a matter of if you have noise in your data and you're trying to cross-validate different portions of the data set or even randomize it, okay, when does that start actually not working? So those are the statistical tests that are run. It's got a robustness against cross-validation. I think, oh, yeah. I knew I wanted to do, I think I have, yeah. So here's some of the specific papers that if you want to look up some of the details, they're in these. The top half is the stem cell, as I said, which was really done in the earlier part of this last decade. And then the bottom set is the hepatocyte work. So if you want to look up the details of the procedures and methods, it's all in here. So I'll leave that up.
Any last questions for Doug?
All right, we're done. Great.